You're sitting in a doctor's office. Not because anything is dramatically wrong. This is a routine visit, the kind where you fill out forms and answer questions, and wait in a room with magazines that are 6 months out of date. The nurse has just taken your blood. Standard procedure. A few vials, a label, off to the lab. You have done this before. You will do it again. You think nothing of it. Then the results come back, and there is a note, a small flag in the corner of your blood work report. 2 letters and a symbol so small you almost miss it. RH negative. And suddenly, you are in a different conversation entirely. Maybe the doctor explains it calmly. About 15% of the population. Nothing to worry about in most circumstances. Relevant in pregnancy. Relevant for transfusions. Keep a medical alert card. Standard medical advice. Delivered with the reassuring competence of someone who has explained this hundreds of times. Or maybe you go home and type those 2 words into a search engine. And then the other conversation begins. The one that is considerably less calm. The one that involves ancient astronauts and reptilian bloodlines and Basque origins and the lost tribes of Israel, and the idea, stated with complete confidence by people who have clearly thought about this for a very long time, that RH negative blood is the mark of a different kind of human being, a purer bloodline, an ancient lineage, possibly extraterrestrial. You close 17 browser tabs and sit back and wonder what just happened. What just happened is that you have stumbled into one of the most fascinating intersections of genuine science, legitimate historical mystery, cultural mythology, and spectacular internet pseudoscience that the modern information landscape has to offer. Because here's the thing about RH negative blood. The real story, the actual documented, peer-reviewed, scientifically established story, is genuinely interesting. Interesting enough that it does not need ancient astronauts or reptilian overlords to be compelling. It involves evolutionary biology, population genetics, the mystery of how a potentially lethal blood incompatibility persisted in human populations, the remarkable geographic distribution of blood type frequencies, the specific puzzle of the Basque people, the history of one of the most consequential medical discoveries of the 20th century, and questions about human prehistory that scientists are still actively working to answer. And then, alongside that genuine story, there is the mythology, the elaborate, creative, occasionally internally consistent body of alternative theories about what RH negative blood means that has developed in the internet era and shows no signs of slowing down. This mythology is interesting in its own right, not because it is true, it is not, but because of what it reveals about the human need to find significance in biological difference, to construct narratives of special origins and hidden identity, to make the accident of blood type into something that means something about who you are and where you come from. This video is going to take both seriously the science, because the science is genuinely remarkable and deserves to be understood clearly, and the mythology, because the mythology tells a story about us, about how human beings process uncertainty, construct identity, and find meaning in the biological lottery of their own bodies. We are going to start at the beginning, the very beginning, with a question that sounds simple and turns out to be anything but. What exactly is RH negative blood? And why does it matter so much? Let's find out. Chapter 1. The blood. What RH negative actually is and why it exists. Let's get the science right first, not in a dry textbook way. In the way that makes you realize the actual biology is more interesting than anything the conspiracy theories invented to replace it. Your blood is not simply red liquid. It is an extraordinarily complex biological system carrying oxygen and nutrients and immune cells and clotting factors, and a remarkable amount of identifying information. On the surface of your red blood cells, there are proteins, antigens in the technical vocabulary, that serve as molecular identity markers. Different people have different combinations of these antigens, and the pattern they form defines your blood type. The ABO blood group system, the one that gives you A, B, AB, or O, is the most famous of these systems, the one that shows up on medical alert bracelets and in those personality quizzes that were popular for a while on social media. It was discovered by Carl Landsteiner in 1901, earning him the Nobel Prize in Physiology or Medicine in 1930. And its discovery transformed medicine by making blood transfusion reliably safe for the first time. But the ABO system is only one of more than 40 blood group systems currently recognized by the International Society of Blood Transfusion. There are antigens on your red blood cells that belong to the Kell system, the Duffy system, the Kidd system, the Lewis system, and many others, each one representing a different set of proteins with different distributions in different populations. The RH system, named for the rhesus macaque monkey for reasons we will get to shortly, is the second most important of all these systems, and the most important after ABO for clinical medicine. It involves a set of proteins encoded by two closely related genes on chromosome 1. The most significant of these proteins, clinically, is the one encoded by the RhD gene, the so-called D antigen, or Rh factor. If you have the D antigen on your red blood cells, if your RhD gene is active and producing the protein, you are Rh positive. This is the most common condition. Approximately 85% of the global population is Rh positive, though this number varies considerably by ancestry and geography, as we will see. If you do not have the D antigen, if your Rhd gene is either absent or non-functional, you are Rh negative. You are missing a protein that the majority of the human population carries. This is the key insight that the mythology tends to invert. Rh negative is not the presence of something extra, it is the absence of something. Rh negative individuals lack a protein that Rh positive individuals have. The question that evolutionary biology has to answer is not where Rh negative blood came from. It is why the absence of this protein persisted in human populations rather than being eliminated by natural selection. And this is where the story gets genuinely interesting. The RhD gene, the gene responsible for producing the D antigen, is believed to have been present in the common ancestor of all modern humans. All primates, in fact, have versions of this gene and the protein it produces. The Rh negative condition, the absence of the functional D antigen, represents a mutation or deletion that occurred at some point in human prehistory, and then, for reasons that are still not entirely clear, spread and persisted in certain human populations rather than being eliminated. Why would a mutation that removes a functional protein persist? Natural selection generally favors functional proteins over their absence. There is usually a reason a protein exists, and losing it tends to have consequences. The fact that Rh negative blood persisted in human populations at frequencies as high as 35% in some groups, rather than being selected out over millennia, suggests that either the absence of the D antigen confers some advantage in certain environments, or that the selective pressure against it is weaker than the selective pressure in the ABO system, or some combination of both. The selective pressure question is complicated by one of the most dramatic medical consequences of Rh negative blood, hemolytic disease of the newborn, also called erythroblastosis fetalis. This condition occurs when an Rh negative mother carries an Rh positive fetus, a situation that arises when the father is Rh positive and the fetus inherits his positive status. If the mother's blood comes into contact with the fetus's blood, typically during delivery or sometimes during pregnancy, the mother's immune system may recognize the D antigen on the fetal red blood cells as foreign and produce antibodies against it. In a first pregnancy, this sensitization is typically not severe enough to cause major problems for the fetus. The immune response develops after delivery, but in subsequent pregnancies with Rh positive fetuses, the mother's immune system already has the antibodies prepared, and it can mount a rapid, powerful response against the fetal blood cells, destroying them and causing severe anemia, organ damage, and death in the fetus or newborn. This is a serious condition. Before modern medicine developed treatments and preventions, specifically Rh immunoglobulin, a preparation of antibodies that prevents sensitization and is given to Rh negative mothers during and after pregnancy. It killed substantial numbers of babies. The historical rate of fetal and infant mortality attributable to Rh incompatibility in populations with significant Rh negative frequency is difficult to estimate, but it was not negligible. So here's the evolutionary puzzle. Rh negative blood creates a condition that can kill the babies of Rh negative mothers who carry Rh positive fetuses. This is a genuine selective disadvantage. It reduces reproductive success. And yet Rh negative blood persisted, and in some populations persisted at very high frequencies. How? Several explanations have been proposed, none of them definitively established. One possibility is that the Rh negative condition confers some other advantage. Resistance to a specific pathogen, perhaps, or some metabolic benefit in certain environments that offsets the reproductive disadvantage of hemolytic disease. Some researchers have proposed that Rh negative individuals may have some degree of resistance to certain parasites, including Toxoplasma gondii, a common parasitic infection. The evidence for this is preliminary and contested. Another possibility is that the selective disadvantage of Rh negative blood is smaller than it appears, because in small, isolated populations where everyone tends to have the same blood type, where an Rh negative woman is more likely to have children with an Rh negative man, producing only Rh negative offspring who are not at risk, the hemolytic disease mechanism never comes into play. If the ancestral population in which Rh negative mutation first arose was small enough and isolated enough that Rh negative individuals mostly mated with other Rh negative individuals, the condition could spread and fix in that population without the reproductive penalty. This last explanation points toward what is genuinely one of the most interesting aspects of the Rh negative story, its extraordinary geographic and population distribution, because Rh negative blood is not distributed evenly across human populations. It is clustered, concentrated in specific populations in specific parts of the world, in a pattern that tells a story about human prehistory that we are still working to fully read. Chapter 2, the map, where Rh negative blood lives and why. Imagine you could color a map of the world according to the frequency of Rh negative blood in each population. Most of the map would be in pale shades. The majority of the world's population is Rh positive, and Rh negative frequencies below 5% are the global norm. East Asia would be nearly white. Rh negative frequency among Han Chinese, Japanese, and Korean populations is roughly 1% or less. Sub-Saharan African populations would be similarly pale, low single-digit percentages. Indigenous American populations, Pacific Islander populations, South Asian populations, all clustering at the low end of the frequency range. Then your eye would travel to Europe, and there the map would darken. European populations generally have Rh negative frequencies between 10 and 18%, substantially higher than most of the world. That's already striking, but then you would notice one region where the color deepens to something much more intense: the western edge of Europe, the Iberian Peninsula, and most dramatically, a specific group of people straddling the border between what is now northern Spain and southern France, the Basques. The Basque people have the highest frequency of Rh negative blood of any population on Earth. Approximately 35% of Basques are Rh negative. In some Basque subpopulations, the frequency may be even higher. This number is so far outside the global norm, more than double the European average, roughly 30 times the East Asian frequency, that it immediately demands explanation. And the explanation turns out to require understanding who the Basques are, because the Basques are themselves one of the most fascinating puzzles in European prehistory. The Basque language, Euskara, is a language isolate. This is the technical term for a language that has no demonstrated relationship to any other known language family. Every other language in Europe belongs to the Indo-European family. English, French, Spanish, German, Italian, Russian, Greek, and dozens of others are all related to each other, descended from a common ancestral language, spoken perhaps 6,000 years ago. Even the non-Indo-European languages of Europe, Finnish, Estonian, Hungarian, are related to each other in the Uralic language family. Basque is related to nothing. It has been spoken in its current location, in the mountains and coastal areas of the Western Pyrenees, for as long as there is any historical record, and its relationship to any other language, living or dead, has resisted the efforts of generations of linguists to establish. It is, in the most literal sense, an orphan language. This linguistic isolation, combined with the extraordinary Rh negative frequency, suggests something important about Basque origins. They are descendants of a population that was in Europe before the Indo-European migrations that spread across the continent beginning roughly 6,000 years ago. They are, in the current best understanding of European population genetics, primarily descended from the early European hunter-gatherers, the people who lived in Europe before farming arrived from the Near East, with relatively little genetic contribution from the subsequent waves of migration that shaped the ancestry of most other Europeans. The early European hunter-gatherers, the people who painted the caves at Lascaux and Altamira, who hunted mammoths across the glacial landscape of Pleistocene Europe, who lived in Europe for tens of thousands of years before the Neolithic agricultural revolution arrived, were apparently the original carriers of high Rh negative frequency in the European population. As farming spread across Europe, bringing new populations from the Near East who had lower Rh negative frequencies, the hunter-gatherer genetic contribution to European populations was diluted. But in the Basque Country, a mountainous, geographically isolated region that may have received the agricultural revolution later and integrated the new arrivals less thoroughly. The original hunter-gatherer genetic signature, including the high Rh negative frequency, was preserved. This is the genuine scientifically supported explanation for the Basque Rh negative anomaly. It is a window into the genetic makeup of pre-Neolithic European hunter-gatherers preserved in a population that has maintained its distinctiveness through geographic isolation and cultural continuity for thousands of years. This is a remarkable story. It does not require aliens. It does not require a secret bloodline. It requires only the ordinary, extraordinary processes of human prehistory: migration, isolation, genetic drift, and the persistence of ancient populations in geographically protected pockets. But let's look at the full global distribution more carefully, because the Basque case is not the only interesting feature of the Rh negative map. The concentration of Rh negative blood in European and specifically Western European populations has led researchers to ask whether there might be environmental factors specific to Europe that favor the persistence of Rh negative blood. One hypothesis involves the specific disease environment of Europe, the particular pathogens that were endemic in the European environment during the long prehistoric period when these blood type frequencies were being established. If Rh negative blood conferred resistance to a pathogen particularly prevalent in Europe, or if Rh positive blood made European populations more susceptible to a European specific disease. This could explain why European populations ended up with substantially higher Rh negative frequencies than populations in other parts of the world. Specific pathogens that have been proposed in this context include the bubonic plague bacterium. Some research has suggested that certain blood type variants may influence susceptibility to Yersinia pestis, the organism responsible for plague, and various parasitic organisms. The evidence remains preliminary, but the direction of the hypothesis is scientifically reasonable. Blood type antigens are, after all, surface proteins that pathogens interact with when they infect cells, and it would not be surprising if different blood type variants conferred different degrees of susceptibility or resistance to different pathogens. The distribution of Rh negative blood outside of Europe is also interesting, though at lower frequencies. Populations in the Canary Islands, specifically the ancient Guanche population, who inhabited the islands before European contact, had Rh negative frequencies comparable to or higher than mainland European populations, despite their geographic position off the coast of Africa. This has been used by various alternative theories to suggest mysterious ancient connections, the Guanche as descendants of Atlanteans, or of ancient Egyptians, or of various other proposed ancient civilizations with elevated Rh negative frequencies. The more mundane explanation is probably that the Guanche were descendants of Berber populations from North Africa, who settled the Canary Islands in antiquity, and that certain North African Berber populations also have elevated Rh negative frequencies, not as high as the Basques, but higher than most African populations. The Berbers, like the Basques, appear to have significant ancestry from pre-agricultural populations, and their higher Rh negative frequencies may reflect, like the Basques, the preservation of an ancestral genetic signature. The geography of Rh negative blood, in other words, is telling us something real and interesting about human prehistory, about the ancient populations of Europe and North Africa, about the patterns of migration and admixture that shaped the genetic landscape of the modern world. It is a legitimate and actively researched area of population genetics, and the picture it is painting becomes clearer with each new ancient DNA study that adds data points to our understanding of who lived where and when, and how they moved and mixed across the landscape. Chapter 3. The Discovery. How the world found out about Rh blood. The The story of how the Rh blood group system was discovered is a story about a monkey, a rabbit, a medical mystery, and a discovery that saved, and continues to save, millions of lives. In 1937, Karl Landsteiner, the same Austrian immunologist who had discovered the ABO blood group system 4 decades earlier, and who was by then working at the Rockefeller Institute in New York, began a series of experiments with his colleague Philip Levine. They were investigating a disturbing medical phenomenon. In a small but consistent percentage of blood transfusions between donors and recipients who were ABO compatible, who should have been safe to transfuse, patients were experiencing severe transfusion reactions, hemolysis, fever, kidney failure, sometimes death. Something was wrong that the ABO system could not explain. There was another antigen in the blood, apparently, that they hadn't identified yet. Meanwhile, Landsteiner and another colleague, Alexander Weiner, were conducting a separate series of experiments involving rhesus macaque monkeys, a species of macaque widely used in biomedical research. They injected rhesus monkey blood into rabbits, provoking the rabbit immune systems to produce antibodies against the foreign blood. They then tested these rabbit antibodies against human blood and found that the antibodies reacted with agglutinated the red blood cells of approximately 85% of humans. The other 15% of human red blood cells were not agglutinated. The protein that the rabbit antibodies were reacting with, the protein present in 85% of humans and absent in 15%, was named the RTRH factor, after the rhesus monkey that had been used to produce the antibodies. The 85% who had it were Rh positive. The 15% who lacked it were Rh negative. It is worth noting that the naming was based on a misunderstanding that took decades to fully correct. The antibodies produced against rhesus monkey blood do not actually react with exactly the same antigen as human Rh antibodies. The rhesus monkey antigen and the human D antigen are similar but not identical. The name Rh factor was retained for the human system despite this, because it had already become established in the medical literature. The discovery of the Rh system was real and correct. The naming connection to rhesus monkeys was technically slightly misleading, but the clinical significance of the discovery was immediately clear, and it solved a mystery that had been causing untold suffering for years before anyone understood what was happening. The mystery was hemolytic disease of the newborn, the condition we described in the previous chapter, in which an Rh negative mother's immune system attacks the red blood cells of an Rh positive fetus. This condition had been observed, described, and puzzled over by physicians for centuries. The ancient medical literature contains descriptions of jaundiced newborns, the yellow color produced by the breakdown products of destroyed red blood cells, and of infants who were born severely ill or who died in the first days of life, from what we now recognize as severe hemolytic disease. Physicians had noticed that the condition tended to affect later-born children more severely than firstborns. They had noticed that it sometimes seemed to run in families. They had noticed that it was associated with jaundice and with severe anemia. But without understanding blood types, they had no framework for explaining what they were seeing and no mechanism for preventing it. Philip Levine, working with Landsteiner on the transfusion reaction problem, made the crucial clinical connection in 1941. He was investigating a case in which a woman had received a blood transfusion that was ABO compatible, but still caused a severe reaction, and who subsequently delivered a severely ill infant. Levine realized that the mother had become sensitized to the Rh factor. She was Rh negative. The transfused blood had been R. RH positive, and her immune system had produced anti-RH antibodies that were then attacking the RH positive blood cells of her infant. This was the key insight: RH sensitization, the development of antibodies against the RH factor, could happen either through transfusion with RH positive blood or through pregnancy with an RH positive fetus. And once sensitized, an RH negative woman's immune system would attack RH positive blood cells wherever it encountered them, including in her own subsequent children. The discovery of this mechanism immediately opened pathways to prevention and treatment. If Rh negative women were given only Rh negative blood in transfusions, they would not be sensitized through that route. If the sensitization during pregnancy could be prevented, if the immune response could be blocked, the disease could be prevented. The development of Rh immunoglobulin, the preventive treatment that is now routinely given to Rh negative pregnant women, was one of the great medical success stories of the 20th century. The preparation containing anti-Rh antibodies derived from donors who had been sensitized to the Rh factor. It works by rapidly clearing any fetal Rh positive red blood cells from the mother's circulation before her immune system has time to mount a primary immune response. Given at 28 weeks of pregnancy and within 72 hours of delivery, it prevents sensitization so effectively that hemolytic disease of the newborn has become extremely rare in countries with good prenatal care. Before your Rh immunoglobulin became available in the 1960s, hemolytic disease of the newborn was a major cause of infant mortality. The numbers are difficult to estimate precisely, but some calculations suggest that tens of thousands of babies died annually in the United States alone from Rh incompatibility before prevention became available. Since then, the routine administration of Rh immunoglobulin has saved hundreds of thousands of lives, perhaps millions globally, and reduced AR, once common tragedy, to a relative rarity. This is the medical legacy of the Rh blood group discovery. Not aliens, not ancient conspiracies. A monkey, a rabbit, a careful scientist, and a series of experiments that led to one of the most effective public health interventions of the 20th century. The story of that discovery is more remarkable than anything the alternative theories have proposed, and it has the considerable advantage of being true. Chapter 4. The Genetics. What your DNA actually says about your Rh negative blood. The molecular genetics of the Rh blood group system are, it turns out, considerably more interesting than a simple positive or negative binary suggests. And understanding the genetics helps explain some of the most puzzling features of the Rh negative story, including why the condition exists at all, why it persists, and what it actually means about a person's ancestry. The Rh blood group system is encoded by two genes on chromosome 1 that are located very close together and are closely related to each other. So closely related that they appear to be the result of a gene duplication event relatively recent in evolutionary terms. These two genes are called RHCE and RHD. The RHCE gene encodes proteins that produce the C, CCE, and E antigens of the Rh system, a set of additional blood group antigens that are clinically relevant, particularly for transfusion medicine, but less prominent in public understanding than the main D antigen. The RHD gene is the one that determines whether you are Rh positive or Rh negative in the conventional sense. If you have a functional RHD gene, you produce the D antigen, and you are Rh positive. If your RHD gene is absent or non-functional, you are Rh negative. Here's what makes this genetically interesting. In most Rh negative people of European ancestry, the RhD gene is simply absent. It has been deleted from the chromosome entirely. A chunk of DNA that includes the entire RhD gene is missing. You are not Rh negative because of a subtle mutation in the gene. You are Rh negative because the gene itself is gone. This kind of deletion, the absence of an entire gene rather than a mutation within it, is actually relatively unusual in the context of blood group genetics, where most variations are produced by point mutations or small changes within genes rather than wholesale deletions. The RhD deletion is large enough and distinctive enough that it can be detected reliably with modern molecular techniques, and its specific characteristics can be used to trace the history of the Rh negative condition in human populations, in African populations with Rh negative phenotype. And as noted earlier, Rh negative frequency is low, but not zero in African populations. The mechanism is often different. Many African Rh negative individuals have an RhD gene that is present but non-functional due to a specific mutation that disables the gene without deleting it. This RhD pseudogene, called RhD star IPSI, is found at relatively high frequency in African populations and represents an independently arising loss of RhD antigen expression that occurred through a different molecular mechanism than the deletion that accounts for most European Rh negative individuals. This is genetically significant because it suggests that Rh negative phenotype, the absence of the D antigen, has arisen multiple times independently in human history through different molecular mechanisms in different populations. This rules out the possibility that all Rh negative people share a single common ancestor who first lost the RhD gene. Rather, the loss of D antigen expression has occurred through at least two independent mutational events in different ancestral populations. The evolutionary question of why these loss of function variants persisted is genuinely unresolved. The deletion is old enough, estimated from molecular clock calculations at perhaps 200,000 to 300,000 years, though these estimates have significant uncertainty that it could date to the time of anatomically modern humans, or even earlier in our ancestral lineage. Ancient DNA studies, the analysis of genetic material extracted from archaeological human remains, have been transforming our understanding of prehistoric population genetics over the past decade, and the Rh blood group is one of the many genetic systems that these studies are illuminating. Ancient European hunter-gatherers whose DNA has been analyzed do appear to have had elevated frequencies of Rh negative genotypes compared to the Neolithic farmers who subsequently spread across Europe. Consistent with the hypothesis that high Rh negative frequency in modern Europeans, and particularly in the Basque population, is a legacy of the pre-agricultural European population. The specific ancient DNA evidence is not yet comprehensive enough to reconstruct the full prehistoric distribution of Rh negative blood with confidence, but the direction of the evidence is consistent. Rh negative blood is older in the European population than the arrival of farming, and its current distribution reflects a process of dilution as farming populations expanded across the continent while preserving the highest frequencies in the populations that maintained the greatest genetic continuity with the pre-farming hunter-gatherers. The genetic story of Rh negative blood is therefore a story about deep time, about genetic variants that have been with humanity for hundreds of thousands of years, that have been carried through the migrations and population replacements and admixtures that shaped the modern human world and that are preserved today in patterns that reflect the complex history of who lived where and when. This is genuinely fascinating science. It is the kind of story that makes the conspiracy theories redundant, not because they are uninteresting, but because the actual story is more interesting, deeper, and more connected to the real history of our species than anything a theory about reptilian bloodlines can offer. But the theories exist, and they have their own logic and their own appeal. And to understand the full story of our H-negative blood, we need to understand them too. Chapter 5. The mythology, where the conspiracy theories come from you, are on the internet. It is approximately 2 a.m. You have been down a rabbit hole for 3 hours. That started with a routine search about blood types and has taken you through a landscape of websites, YouTube videos, forum threads, and self-published books that has become progressively more elaborate and more confident as the night has worn on. You have learned, or at least been told, with the specific insistence of the internet at 2 a.m., that Rh-negative blood is the mark of a non-human bloodline, that it cannot have evolved naturally, that it is the genetic signature of the Nephilim, the fallen angels described in Genesis, or of extraterrestrial visitors who interbred with early humans, or of the survivors of Atlantis, or of an ancient race of beings who preceded modern humanity and whose descendants are among us still, identifiable by their blood type, their green or hazel eyes, their tendency toward psychic sensitivity, and their inexplicable sense of always having been somehow different from everyone around them. The websites are very sure about all of this. The certainty is, in fact, one of the more striking features of the genre. How did we get here? How did a genuinely interesting piece of immunology become the substrate for one of the more elaborate mythological systems of the internet age? The answer involves several converging streams of cultural history that are worth tracing, because understanding where the mythology comes from helps us understand both why it is wrong and why it is so persistently appealing. The first stream is the oldest, and it predates the internet by millennia. It is the human tendency to find significance in blood, to treat blood as the carrier of identity, ancestry, spiritual essence, and divine connection. This tendency is so universal and so deep in human culture that it appears in virtually every religious and mythological tradition ever documented. Blood covenants, blood sacrifices, blood as the seat of the soul, blood as the medium of divine communication. The idea that your blood tells you something essential about who you are and where you come from is not a modern invention. It is one of the oldest ideas in the human repertoire. When modern medicine discovered, in the early 20th century, that blood types were real and heritable and varied between individuals and populations, it handed this ancient tendency a new set of tools. The discovery of the ABO system was almost immediately caught up in the racial science of the early 20th century. Researchers in the 1920s and 1930s attempted to use blood type frequencies to distinguish between racial groups, to trace racial migrations, and to establish hierarchies of racial purity. This project was scientifically confused and morally catastrophic, but it established the template of treating blood type as a marker of deep racial or ancestral identity that has persisted in various forms to the present. The specific Rh-negative mythology that circulates today draws on several distinct sources that have been combined in the creative ecosystem of internet culture into a reasonably coherent alternative history. Let's trace the main currents. The Nephilim connection is the oldest and theologically richest strand. The Nephilim appear in Genesis 6:1-4, in one of the most cryptic passages in the Hebrew Bible. When men began to multiply on the face of the ground, and daughters were born to them, the sons of God saw that the daughters of men were fair, and they took to wife such of them as they chose. The Nephilim were on the earth in those days, and also afterward, when the sons of God came in to the daughters of men, and they bore children to them. This passage has been the subject of intense theological debate for millennia. Who were the sons of God? Who were the Nephilim? The most common interpretation in Jewish and early Christian tradition was that the sons of God were fallen angels, and the Nephilim were their hybrid offspring, beings of mixed human and angelic blood who walked the earth before the flood. The physical descriptions of the Nephilim in various ancient texts tend toward the extraordinary, giants, mighty men, beings of unusual power and appearance. The connection of this ancient tradition to Rh-negative blood was apparently made in the late 20th century by writers working in the emerging genre of alternative history and ancient astronaut theory. The reasoning goes like this. If the Nephilim were the offspring of non-human beings and human women, they would have had blood that was unlike ordinary human blood. The Rh incompatibility between Rh-negative mothers and Rh-positive fetuses. The fact that an Rh-negative mother's body can react against an Rh-positive fetus as if it were foreign is interpreted in this framework as the body recognizing that the fetus carries blood from a different lineage. The Rh-negative person's body knows at an immunological level that the Rh-positive blood is foreign. This is taken as evidence that Rh-positive blood is the contaminated blood, the human blood that was altered by interbreeding with something non-human. And Rh-negative blood is the original, pure, pre-contamination human blood. This interpretation gets the immunology precisely backwards. It is the D antigen present in Rh-positive blood that is the original primate condition, and its absence in Rh-negative individuals that represents the derived mutation. But it has the narrative elegance of reversing an apparent disadvantage, the Rh incompatibility, into evidence of special origin. The Rh-negative mother's body isn't experiencing a medical complication. She is recognizing foreign blood. The ancient astronaut strand, associated primarily with writers like Zecharia Sitchin, Erich von Däniken, and their numerous successors and online interpreters, takes a parallel route. In the ancient astronaut framework, which achieved mass cultural penetration through the History Channel series, Ancient Aliens, beginning in 2010, various ancient textual references to gods, angels, and divine beings are reinterpreted as descriptions of extraterrestrial visitors. The Anunnaki of Sumerian mythology, a pantheon of major deities, are reinterpreted as aliens who visited Earth, created or modified humanity through genetic engineering, and left behind various traces in human culture and biology. Rh-negative blood fits into this framework as the genetic legacy of the alien visitors themselves, or of their hybrid offspring, persisting in the human population as a marker of non-terrestrial ancestry. The specific claim varies by source. Some versions hold that Rh-negative blood indicates pure alien ancestry, others that it marks the descendants of alien-human hybrids, others that it is simply the original genetic template that the alien engineers used, preserved in certain populations, while others were modified. The claim that Rh-negative blood could not have evolved naturally, a statement that appears with remarkable frequency in this literature, is the linchpin of the argument. If Rh negativity cannot be explained by ordinary evolutionary processes, then it must have an extraordinary explanation. And the extraordinary explanation on offer is extraterrestrial origin. The argument requires the premise to be true, that evolutionary biology genuinely cannot explain the existence and distribution of Rh-negative blood. As we have seen in the previous chapters, this premise is false. Evolutionary biology has entirely plausible explanations for the existence and distribution of Rh-negative blood. Explanations that are consistent with what we know about the molecular genetics of the RhD gene, the population genetics of pre-agricultural European hunter-gatherers, and the specific history of the Basque population. These explanations have gaps and uncertainties, because that is the nature of science dealing with deep prehistory, but they are not failures. They are the normal, productive uncertainty of a research field that is actively generating new evidence and refining its models. The mythology, by contrast, presents certainty where there is none and treats the gaps in scientific knowledge as proof of its own preferred explanation rather than as invitations to continued inquiry. The third major strand of Rh-negative mythology is the reptilian bloodline theory, most associated with the British author David Icke, whose books beginning in the early 1990s developed an elaborate alternative history in which shape-shifting reptilian beings from another dimension have infiltrated human society and are represented in significant numbers among the global elite. This theory is the most explicitly conspiratorial strand of Rh-negative mythology, and it shares some features with anti-Semitic conspiracy theories, a connection that has been noted and criticized by scholars of conspiracy culture. In Icke's framework, certain bloodlines, particularly those of European royal families and global power elites, carry reptilian DNA that can be identified through various means, including blood type. Rh-negative blood appears in some versions of this theory as a marker of the reptilian bloodline itself, or as a marker of human bloodlines that have been targeted for particular attention by the reptilian elite. The specific mechanics vary between different iterations of the theory, and Icke's own claims have evolved over time. The reptilian bloodline theory has achieved a cultural footprint far out of proportion to its plausibility, partly through Icke's prolific writing and speaking career, and partly through the viral dynamics of internet culture, where its combination of the familiar, celebrity conspiracy theories, and the bizarre, actual reptilians makes it highly shareable regardless of its truth value. What all three strands of Rh-negative mythology share: the Nephilim connection, the ancient astronaut theory, and the reptilian bloodline theory, is a fundamental structure. The idea that Rh-negative blood is a marker of non-ordinary human ancestry, that it separates its bearers from the majority of humanity in ways that are spiritually, genetically, or cosmically significant, and that this significance has been suppressed or ignored by mainstream institutions. This structure has a powerful psychological appeal that we will examine in the next chapter. But first, let's look at some of the specific empirical claims that the mythology makes, because some of them can be directly evaluated against the scientific evidence. And the evaluation is instructive. The claim that Rh-negative people tend to have green or hazel eyes, red or auburn hair, lower than average body temperature, higher than average blood pressure, higher than average IQ, enhanced intuition or psychic ability, and an inexplicable sense of not fitting in with society. This cluster of alleged characteristics is one of the most widely circulated elements of Rh-negative mythology, appearing in slightly different versions across hundreds of websites and forum posts. Let's be direct. There is no scientific evidence for any of these correlations. Eye color, hair color, body temperature, blood pressure, intelligence, and psychological characteristics are all determined by genetic factors that are entirely independent of the RHD gene. The genes that influence eye pigmentation are on different chromosomes from the RHD gene. The genes that influence hair pigmentation are similarly unrelated. There is no known biological mechanism by which the presence or absence of the D antigen on red blood cells would influence any of these characteristics. The clusters of characteristics described in Rh-negative mythology appear to be assembled from a combination of several sources. Genuine stereotypes about European populations with high Rh-negative frequencies, who do tend, for entirely independent reasons, to have lighter eye and hair color than global averages. Post hoc pattern recognition. People who read that Rh-negative individuals tend to have certain characteristics and then notice those characteristics in themselves. And the general human tendency to accept descriptions of special qualities when they are framed as applying to a group we belong to. The Barnum effect, the same psychological mechanism that makes horoscopes feel personally accurate. The inexplicable sense of not fitting in is particularly worth examining because it appears so consistently and resonates so powerfully with so many people who encounter Rh-negative mythology. This sense of being somehow different, of not quite belonging to the ordinary human world, of having experiences and perceptions that others don't share, is not a specific characteristic of Rh-negative individuals. It is a characteristic of a very large proportion of the human population, for a very wide range of reasons. The experience of feeling different is so common as to be, paradoxically, universal. The appeal of a biological explanation for this feeling, one that reframes feeling like an outsider as evidence of special ancient lineage rather than ordinary human alienation, is obvious and understandable, even if the explanation is wrong. Chapter 6. The Basques Revisited. What the oldest Europeans actually tell us. We need to spend more time with the Basques because they are simultaneously the most scientifically interesting population in the Rh-negative story and the most thoroughly mythologized one. The genuine scientific puzzle of Basque origins, which is real, fascinating, and only partially resolved even by modern genetic research, has attracted an enormous overlay of alternative theories that range from the linguistically creative to the geologically improbable. The Atlantis connection is perhaps the most enduring of the non-scientific Basque origin theories. The idea, stated in various forms by various authors over the past 2 centuries, goes roughly as follows. Atlantis was a real civilization located in the Atlantic Ocean, approximately where the Azores or the Canary Islands now are. When Atlantis sank, in the cataclysm described by Plato, its survivors fled to the nearest land. The coast of Western Europe and North Africa was closest. The Basques are the descendants of the Atlantean survivors, which is why their language is unrelated to any other language in Europe, and why their blood type frequencies are anomalous. This theory has a certain narrative elegance. It explains 2 genuine anomalies: the linguistic isolate status of Basque and the anomalous Rh-negative frequency, with a single dramatic historical event. It also connects to the most famous lost civilization myth in Western culture, giving the Basques a romantic, ancient heritage that is considerably more exciting than descendants of pre-Neolithic hunter-gatherers who happen to live in a geographically isolated area. The problems with the Atlantis theory are, unfortunately geological, archaeological, and genetic simultaneously. There is no geological evidence for a large Atlantic landmass that sank within human prehistory. The Atlantic Ocean floor has been extensively mapped, and there is nothing there that resembles a submerged continent of the scale Plato described. Plato's own account of Atlantis, in the dialogues Timaeus and Critias, is almost certainly a philosophical fiction or a heavily mythologized account of a real but much more modest historical event, possibly the eruption of Thera and the decline of Minoan civilization. And the modern genetic evidence for Basque origins, which we have already described, places them clearly within the range of Western European hunter-gatherer ancestry, without any signal of a dramatically different or geographically distant origin. The Lost Tribe of Israel connection is another persistent alternative theory for Basque origins. In this framework, the Basques are the descendants of one of the 10 lost tribes of Israel, the northern tribes who were deported by the Assyrian Empire in 722 BC and whose subsequent fate is unknown. The Hebrew-Basque language connection theories claims that Basque and ancient Hebrew share vocabulary and grammatical features that indicate a common origin have been proposed and refuted multiple times. Modern linguistics has found no credible evidence for any such connection. The Basque language is genuinely without demonstrated relatives, which means it cannot be connected to Hebrew any more than it can be connected to any other known language family. The genuinely scientific story of Basque origins, as it has been reconstructed by modern genetics and archaeology, is both more modest and more interesting than any of the alternative theories. The genetic evidence drawn from ancient DNA studies of European prehistoric populations conducted over the past decade shows that the Basque population is primarily descended from a mixture of two source populations: the Western European hunter-gatherers who inhabited Europe during the Paleolithic and Mesolithic periods, and the early European farmers who arrived from the Near East with the spread of agriculture during the Neolithic period. This is, in broad outline, the same ancestral mixture that most modern Europeans descend from, with the addition of steppe ancestry from later Bronze Age migrations. What makes the Basques unusual is the proportions. Compared to most other modern European populations, Basques have relatively more hunter-gatherer ancestry and relatively less steppe ancestry from the later Bronze Age migrations, the migrations that brought Indo-European languages to Europe, and that are thought to be responsible for the spread of most European languages. This reduced steppe ancestry is probably related to the Basque language's survival. The Bronze Age migrations that spread Indo-European languages across Europe did not penetrate the Basque region as thoroughly as they penetrated most other areas, which is why the pre-Indo-European Basque language survived while other pre-Indo-European European languages were replaced. The high Rh negative frequency in Basques is consistent with this ancestry profile. The Western European hunter-gatherers appear to have had high Rh negative frequencies, and populations with more hunter-gatherer ancestry tend to have higher Rh negative frequencies than populations with more Neolithic or Bronze Age farmer ancestry. The Basques, with their elevated hunter-gatherer genetic component, simply preserved more of the ancestral European Rh negative frequency than most of their neighbors. This explanation is, let us be honest, less dramatically satisfying than the Atlantis theory. They have old genetic ancestry from pre-farming Europeans does not have the same narrative punch as they are the survivors of a sunken civilization, but it has something the Atlantis theory lacks. It is supported by actual evidence from actual ancient DNA extracted from actual prehistoric European skeletons. The ancient DNA revolution in archaeology has been one of the most significant developments in the study of human prehistory in the past decade, and it is continuously refining our understanding of where modern populations came from. For the Basques specifically, and for the Rh negative story more broadly, it is providing a framework that is both scientifically credible and genuinely illuminating about the deep history of the people who carry this blood type. The Basques are not Atlanteans. They are not aliens. They are not the lost tribe of Israel. They are something more interesting than any of those things. They are a living genetic window into the world of pre-agricultural Europe, into the people who lived in Western Europe before farming arrived and transformed the population, before the Bronze Age migrations brought Indo-European languages and new genetic lineages, before the sequence of historical events that shaped the modern European genetic landscape. In their blood, and specifically in the high frequency of Rh negative blood, that is the most dramatic expression of their ancestral heritage. They carry a genetic signature that is 10,000 or more years old. That is, if you think about it clearly, considerably more remarkable than being descended from Atlantis. Chapter 7, The Psychology. Why people believe. In the special blood, let's do something that most videos about conspiracy theories avoid, because it requires a degree of empathy for the people who hold the beliefs being examined. Let's try to understand why the Rh negative mythology is appealing, not in the patronizing sense of poor confused people can't evaluate evidence properly. In the genuine sense of what human needs does this mythology serve, and why are those needs so powerful that millions of people find it more compelling than the scientific explanation? Because the numbers are real. The Rh negative mythology has a substantial and devoted following. There are Facebook groups with hundreds of thousands of members. There are YouTube channels with millions of views. There are self-published books, websites, forums, and podcast communities devoted to exploring the significance of Rh negative blood with an enthusiasm and a sense of shared identity that is sociologically genuinely interesting. The first thing to understand is that the appeal is not primarily intellectual. The Rh negative mythology does not succeed because it is more logically coherent than the scientific explanation. It is not, and most of its adherents are not primarily motivated by a failure to understand the science. It succeeds because it meets emotional and social needs that the scientific explanation does not. The most powerful of these needs is the need for a coherent identity narrative. Human beings are story-making creatures. We do not simply experience our lives as a sequence of events. We need to understand those events as a story, with a coherent thread connecting past to present, with an explanation of why we are who we are and where we came from. Blood type is a biological given. You were born with it. It will never change. It is part of you in the most literal biological sense. The appeal of a mythology that makes your blood type. The key to a deeper story about your identity and your origins is enormous. And for Rh negative individuals, that story is particularly attractive because it transforms what might otherwise seem like a medical complication. A blood type that creates potential problems in pregnancy and requires special consideration and transfusion into a mark of distinction. The Rh negative mythology does not frame Rh negative blood as an absence, as the science does. It frames it as a presence, a special quality, an ancient inheritance, a marker of uncommon lineage. This is a deeply satisfying inversion of the medical narrative, which simply describes Rh negative people as lacking a protein that most people have. The second powerful need that the mythology meets is the need to explain the sense of difference. As we noted earlier, the feeling of not quite fitting in, of being somehow different from the people around you, of having perceptions or experiences or values that set you apart is extremely common. It is also extremely uncomfortable, particularly in a culture that simultaneously celebrates individualism and demands conformity in very specific ways. The Rh negative mythology offers an explanation for this feeling that is both flattering and exonerating. You feel different because you are different, not because of ordinary psychological variation, not because of the universal human experience of being an individual in a social world, but because you carry ancient non-human DNA that makes you genuinely biologically distinct. The alienation is not a problem to be managed or understood. It is evidence of special origin. This framing has the additional advantage of creating a community. The millions of people who identify with the Rh negative mythology are not a random collection of individuals who happen to share a blood type. They are, in the mythology's framing, a people, a lineage, a bloodline, a community of shared origin and shared experience. The Facebook groups and YouTube comment sections and forum threads are not just places to discuss blood types. They are places where people who feel different can find each other, validate each other's experience, and build a shared identity around the biological fact of their blood type. This community building function is one of the most socially significant aspects of the Rh negative mythology, and it is the function that makes it most resistant to debunking. Telling someone that their blood type does not actually make them special is not just making a factual claim. It is challenging the foundation of a community they belong to and an identity they have built. The social cost of accepting the scientific explanation, of leaving the community, of giving up the special identity is substantial, and for many people, it outweighs the epistemic benefits of accuracy. The third need the mythology meets is the need for a grand explanatory framework, a way of understanding not just personal identity, but the larger patterns of history and power. The versions of the Rh negative mythology that involve suppression of knowledge about the special bloodline, that suggest the powers that be know about the significance of Rh negative blood, and are hiding it, connect the blood type to the broader conspiracist narrative about hidden power and suppressed truth that is one of the most persistent and most appealing frameworks for making sense of a complex and often confusing world. Conspiracy theories, as scholars of the genre have noted, are not simply beliefs about specific hidden plots. They are ways of explaining why the world doesn't make the kind of sense it should, why power seems unaccountable, why suffering seems undeserved, why institutions seem to fail the people they are supposed to serve. The grand conspiracy provides an explanation that restores a kind of sense to the world. Things are the way they are not because of the messy, contingent, impersonal processes of history, but because powerful hidden agents have arranged them that way for specific purposes and with specific intentions for our H negative mythology adherents. The conspiracy framing serves a specific function. It explains why the knowledge of the special bloodline is not mainstream. If the knowledge were simply wrong, it might be corrected by evidence. But if it is being suppressed by powerful interests, if the mainstream academic and medical establishment is hiding the truth about Rh negative blood for reasons of their own, then the absence of mainstream confirmation becomes paradoxically confirmation of its own kind. The silence of the experts is taken as evidence that the experts know more than they are saying. This epistemic move treating the absence of evidence as evidence of suppression, is one of the most powerful rhetorical tools in the conspiratorial toolkit, and it is essentially unfalsifiable. Any evidence against the theory can be reinterpreted as disinformation. Any absence of supporting evidence can be reinterpreted as suppression. The theory becomes immune to disconfirmation. Understanding these psychological and social dynamics does not mean that the people who hold these beliefs are unintelligent or irrational in some global sense. They are, for the most part, people with the same cognitive capacities as everyone else, applying those capacities in a social and informational environment that makes certain kinds of errors more likely. The internet, with its algorithmic tendency to serve users content that confirms their existing inclinations, creates particularly fertile conditions for the development and maintenance of alternative belief systems. The Rh negative mythology is, in significant part, an internet phenomenon, shaped by the specific dynamics of how information spreads and how communities form in online environments. Chapter 8. The Celebrity Bloodlines, Famous Rh Negatives, and What They Don't Prove. One of the more creative contributions of the Rh negative mythology to alternative history is the construction of lists, extensive, confident, regularly updated lists of famous historical figures, celebrities, and powerful individuals who are alleged to be Rh negative, and whose Rh negative status is taken as confirming the theory that Rh negative blood is associated with unusual intelligence, creativity, or power. These lists are worth examining because they illustrate several important epistemological problems simultaneously. The famous names that appear most consistently in Rh negative mythology include John F. Kennedy, Abraham Lincoln, Queen Elizabeth II, Barack Obama, Mick Jagger, Paul McCartney, Prince Charles, Albert Einstein, Nikola Tesla, and various other historical and contemporary figures chosen broadly for their prominence and their perceived extraordinariness. The lists vary by source and are updated as new famous. People are identified as putatively Rh negative. There are several problems with these lists, even before we get to the question of whether the people on them are actually Rh negative. The first problem is that the blood types of most historical figures are simply not known. Albert Einstein's blood type is not a matter of public record. Nikola Tesla's blood type is not documented. The various claims that circulate about the blood types of historical figures are, in almost all cases, not based on documented evidence. They are either fabricated, extrapolated from ancestry, which is not a reliable way to determine blood type, since blood type is heritable but not deterministically so, or based on medical records that are not publicly accessible, when actual documented blood types of public figures are available. And the blood types of some political figures have been made public in the context of medical disclosures. The results do not show any particular clustering of Rh negative blood among the famous and powerful. The distribution, such as it is, appears consistent with what you would expect from the general population. The second problem is selection bias. The Rh negative mythology lists contain famous individuals who are alleged to be Rh negative. They do not contain, by definition, the far larger number of famous individuals who are Rh positive. If you make a list of famous and remarkable people of a given blood type, the list will inevitably look impressive because you are selecting from a very large pool of remarkable people and including only those who match your criterion. This tells you nothing about whether that blood type is associated with remarkableness. The third problem is the base rate. Approximately 15% of the global population is Rh negative. somewhat higher in European ancestry populations. Among the many thousands of individuals who achieve historical prominence in science, politics, arts, and other fields, 15% can be expected to be Rh negative by chance alone, without any special connection between blood type and achievement. The existence of famous Rh negative people is therefore not evidence of a connection between Rh negative blood and extraordinary capacity. It is exactly what you would expect. The royal family connection, the claim that the European royal families have a disproportionate prevalence of Rh negative blood, and that this explains their power and their insistence on maintaining dynastic bloodlines, is particularly elaborate and particularly instructive. European royal families did, historically, practice endogamous marriage, marrying within a small pool of related royal families, which had various genetic consequences, the most famous being the spread of hemophilia through the descendants of Queen Victoria. Whether this endogamy resulted in any particular clustering of blood types in the European royal families is not documented in any publicly available medical records, and the genealogical arguments used to suggest it involved too many uncertain steps to be reliable. The actual documented hemophilia in the British royal family's descendants, a condition caused by a mutation in a clotting factor, gene entirely unrelated to blood type, is sometimes confused with or connected to Rh blood type issues in alternative history accounts, apparently because both involve blood and both are hereditary. They are, in fact, entirely unrelated conditions caused by entirely unrelated genes. The connection between them exists only in the creative synthesis of alternative history, not in biology. The political power angle of the royal bloodline argument is worth addressing directly because it raises the question of why, if Rh negative blood were genuinely associated with unusual intelligence or leadership capacity, the institutions of power would want to suppress this knowledge rather than celebrate it. The suppression narrative requires the powerful, who are alleged to be predominantly phrase Rh negative, to be hiding the evidence of their own special status. This is a somewhat unusual conspiracy, in which the conspirators are hiding evidence of their own superiority rather than evidence of their crimes. The internal logic is not impossible, but it requires assuming that the powerful prefer hidden advantage to acknowledge superiority, which runs against most observations of how actual powerful people behave. Chapter 9. The Science Fights Back. What modern research actually shows you are in a genetics laboratory. The year is 2023. Around you, machines the size of refrigerators are humming with a quiet efficiency that belies the extraordinary complexity of what they are doing. Reading, base by base, the genetic sequences encoded in DNA extracted from samples that arrived this morning from a repository of ancient skeletal material excavated from a Mesolithic site in western Iberia. The samples are approximately 8,000 years old. The people they came from were alive in the period before farming reached the Iberian Peninsula. They were hunter-gatherers living on the Atlantic coast of what is now Portugal, fishing and hunting and gathering in a landscape that looked nothing like the modern world. And from their ancient bones, preserved in the right conditions for 8 millennia, you are extracting genetic information that will tell you, among many other things, what blood types they carried. This is real. This is happening. Ancient DNA research, the extraction and analysis of genetic material from archaeological human remains, has transformed our understanding of human prehistory faster than almost any other scientific development of the past 2 decades. It is directly relevant to the Rh negative story because it is providing empirical data about the distribution of blood type variants in prehistoric human populations that was simply unavailable to previous generations of researchers. The results of ancient DNA studies relevant to the Rh negative question have been accumulating for several years. And while the picture is not yet complete, ancient DNA research is expensive, technically demanding, and dependent on the preservation conditions of the skeletal material. The emerging pattern is consistent and illuminating. Western European hunter-gatherers, the people who lived in Europe before the arrival of farming from the Near East beginning approximately 9,000 years ago, do appear to have had elevated frequencies of Rh negative genotypes compared to the farming populations that subsequently spread across Europe. Ancient DNA from Mesolithic sites in Iberia, France, Germany, and Scandinavia shows Rh negative genotype frequencies substantially higher than modern European averages in some sites, consistent with the hypothesis that the ancestral European hunter-gatherer population was the original source of high Rh negative frequency in Europe. The Neolithic farmers who spread agriculture across Europe, originating from populations in the Near East and Anatolia, carried lower Rh negative frequencies, again consistent with the modern pattern, in which populations of Near Eastern and South Asian ancestry tend to have lower Rh negative frequencies than Europeans. As farming spread across Europe and the incoming farming populations mixed with the existing hunter-gatherers, the Rh negative frequency in most European populations was diluted toward an intermediate value. The Bronze Age steppe migrations, the movement of populations from the Pontic-Caspian steppe into Europe, beginning approximately 5,000 years ago, bringing the ancestral Indo-European languages and contributing substantially to the ancestry of most modern Europeans, added another layer to this mixture. The steppe populations also had relatively lower Rh negative frequencies than the hunter-gatherers, further diluting the ancestral high Rh negative frequency signal in most European populations. What this three-layer model of European population history predicts is exactly what we observe. The populations with the most hunter-gatherer ancestry should have the highest Rh negative frequencies, while populations with more farmer and steppe ancestry should have lower frequencies. The Basques, with their elevated hunter-gatherer genetic component and reduced steppe ancestry, fit perfectly at the high-frequency end of this spectrum. This is not speculation. It is a prediction from a model of European population history that is supported by ancient DNA evidence from hundreds of prehistoric individuals across the continent, tested against the modern blood type frequency data, and found to be consistent. The scientific explanation for high Rh negative frequency in European populations, particularly in the Basques, is not a just-so story invented to counter the mythology. It is a rigorously tested model that makes specific predictions and finds confirmation in the data. The ongoing ancient DNA research is continuously refining this picture. Each new ancient genome that is analyzed adds a data point that can confirm, refine, or potentially challenge the existing model. The field is moving fast. A decade ago, it was barely possible to extract reliable ancient DNA from samples older than a few thousand years. Today, researchers are routinely analyzing genomes from individuals who lived 40,000 years ago. The Rh blood group, along with other blood type systems, is increasingly being incorporated into the standard analysis of ancient genomes, building a database of prehistoric blood type frequencies that will eventually give us a much more complete picture of how these variants were distributed across time and space. One particularly interesting area of active research involves the question of what selective pressures, if any, have influenced the frequency of Rh-negative blood in human populations. The neutral model that Rh-negative frequency is simply the result of genetic drift in small isolated populations with no particular selective advantage or disadvantage is possible but may not be the complete story. Several research groups are actively investigating potential selective effects. The most rigorously studied potential selective factor involves infectious disease. Blood type antigens are, by their nature, surface proteins on red blood cells, proteins that interact with the external environment, including the pathogens that infect the body. It is biologically plausible that different blood type variants might interact differently with specific pathogens, conferring different degrees of susceptibility or resistance. For the ABO blood group system, this has been reasonably well established. Type O individuals have some degree of resistance to severe malaria caused by Plasmodium falciparum. Type A individuals have elevated susceptibility to certain bacterial infections, and so on. For the Rh blood group, the evidence is less established, but not absent. A research group studying Rh blood group and infectious disease found preliminary evidence suggesting that Rh-negative individuals may have some difference in susceptibility to Toxoplasma gondii, a common parasitic protozoan that infects a substantial fraction of the global human population. The study found that Rh-negative individuals who had been infected with Toxoplasma showed different behavioral patterns compared to Rh-positive infected individuals. This is a preliminary finding that requires replication and has been contested, and it does not establish that Rh-negative status confers overall resistance to Toxoplasma, but it suggests that the Rh blood group system may have interactions with specific pathogens that could be relevant to understanding why different frequencies of Rh-negative blood have persisted in different populations. Research into the Rh blood group system continues at a fundamental level as well. The molecular structure of the Rhd protein, the protein absent in Rh-negative individuals, has been of interest to cell biologists for reasons that go beyond blood group serology. The Rh proteins appear to play a role in the transport of gases, specifically carbon dioxide and ammonia, across the red blood cell membrane. If the Rhd protein contributes to gas transport efficiency in red blood cells, the absence of this protein in Rh-negative individuals might have subtle physiological consequences that have not yet been fully characterized. This is speculative. The functional role of the Rh proteins is still an active area of research, but it suggests that the biology of the Rh blood group system may be more complex and more physiologically significant than its role as a blood group antigen alone would suggest. The medical management of Rh incompatibility continues to be an active area of clinical research as well. Despite the dramatic success of Rh immunoglobulin in preventing sensitization, not all Rh-negative pregnant women in the world have access to Rh immunoglobulin. It requires refrigerated storage, professional administration, and a functioning healthcare system. In low-resource settings, hemolytic disease of the newborn remains a significant cause of infant mortality, and research into longer-acting, more stable, or more easily administered forms of Rh immunoglobulin is ongoing. The global health dimension of the Rh story is thus not purely historical. It is a continuing challenge, and the scientific community is continuing to work on it. The genetics of Rh blood group variation is also more complex than the simple positive-negative binary suggests. There are dozens of Rh blood group antigens beyond the main D antigen, and the clinical significance of these additional antigens for transfusion medicine, for neonatal medicine, for organ transplantation is an active area of blood bank medicine. Extended Rh genotyping, the molecular determination of an individual's complete Rh antigen profile, beyond just D positive or D negative, is increasingly used in clinical practice, particularly for patients who require repeated transfusions, because precise matching of donor and recipient Rh antigens reduces the risk of alloimmunization and transfusion reactions. The science of the Rh blood group system is, in short, a living field of active research, generating new knowledge about human prehistory, human evolution, infectious disease, susceptibility, red blood cell physiology, and clinical medicine simultaneously. It does not need mythology to be interesting. It is interesting on its own terms, and it is getting more interesting as the research progresses. Chapter 10: The Global Health Story. Rh negative in the developing world. Let's shift focus for a moment from the mythology and the genetics to the part of the Rh negative story that is, in practical human terms, the most important and the least discussed. While the internet debates whether Orsaphia Rh negative blood is evidence of alien ancestry, and while academic researchers trace the prehistoric migrations that shaped blood type distributions, a quiet and ongoing public health challenge is unfolding in hospitals and clinics across the developing world. It is a challenge that kills tens of thousands of babies every year. It is entirely preventable given the right medical resources, and it is not receiving anything close to the attention it deserves. Hemolytic disease of the newborn, the condition caused by Rh incompatibility between mother and fetus, was effectively conquered in wealthy countries in the 1970s and 1980s through the routine administration of RRH immunoglobulin to RRH negative pregnant women. In the United States, the United Kingdom, Western Europe, Australia, and other high-income countries, hemolytic disease of the newborn due to Rh incompatibility is now rare enough that most obstetricians practicing today have never managed a severe case. In low and middle-income countries, the picture is dramatically different. Rh immunoglobulin requires cold chain storage. It must be kept refrigerated from production to administration, and it must be given at specific points during and after pregnancy by trained healthcare providers. It is relatively expensive compared to many other essential medicines. In health systems with limited infrastructure, limited trained personnel, and limited budgets, the routine administration of Rh immunoglobulin to all Rh negative pregnant women is not always achievable. The result is that hemolytic disease of the newborn continues to be a significant cause of fetal and infant mortality in many parts of the developing world. Global estimates of the burden are imprecise. The condition is inconsistently diagnosed and inconsistently recorded in low-resource settings, but some analyses suggest that tens of thousands of babies die annually from Rh incompatibility worldwide, with the vast majority of these deaths occurring in South Asia, Sub-Saharan Africa, and parts of Latin America. The populations most affected are not necessarily the populations with the highest Rh negative frequencies. While Rh negative frequency is lower in South Asia and Sub-Saharan Africa than in Europe, it is not zero. And in large populations, even low-frequency blood type variants affect substantial numbers of people. A 1% Rh negative frequency in a population of 1 billion people means 10 million Rh negative individuals. A fraction of those individuals will have pregnancies with Rh positive partners, will carry Rh positive fetuses, and will be at risk of sensitization. Without Rh immunoglobulin, a fraction of those sensitized mothers will go on to have subsequent pregnancies severely affected by hemolytic disease. The numbers add up. The tragedy is real. It is ongoing, and it is preventable. Organizations working on global maternal and neonatal health have been increasingly aware of this issue. The World Health Organization includes Rh immunoglobulin on its essential medicines list. The list of medicines considered the most important for basic health systems worldwide. But inclusion on the essential medicines list does not ensure availability. Supply chains, regulatory approval, local procurement practices, and financing all affect whether a medicine actually reaches the patient who needs it. Point-of-care blood typing, the ability to determine a patient's blood type quickly and accurately at the bedside or in a clinic without sophisticated laboratory equipment, is one of the key bottlenecks in addressing the global burden of Rh incompatibility. If you cannot reliably identify which pregnant women are Rh negative, you cannot target Rh immunoglobulin to those who need it. In settings with limited laboratory capacity, blood typing may not be available for all pregnant women, or may be inaccurate due to reagent quality or technical issues. Several research groups and global health organizations are working on improving point-of-care blood typing for low-resource settings. Developing paper-based lateral flow tests, smartphone-based imaging systems for reading agglutination reactions, and other low-cost, robust technologies that can reliably determine blood type in field conditions. These efforts are not glamorous. They do not attract the attention that gene therapy or mRNA vaccines attract, but their potential impact on infant mortality is substantial. The financing of Rh immunoglobulin for low-income countries is also an active area of advocacy. The cost per dose of Rh immunoglobulin has been falling as the market has expanded and as biosimilar versions of the preparation have been developed, but it remains above what many low-income country health systems can afford to purchase for all eligible patients. Global health financing mechanisms, including the global health funds, and mechanisms that support access to other essential medicines, have been increasingly engaged on this issue. The story of Rh immunoglobulin is, in global health terms, a story of profound inequality. The technology to prevent hemolytic disease of the newborn has existed for 50 years. It works. It is safe. It is, in the context of what it prevents, not particularly expensive. And yet the condition it prevents continues to kill babies in the parts of the world that can least afford the loss. This is not a medical problem. It is solved medically. It is a health system, economic, and political problem of the kind that global health consistently struggles with. The fact that this ongoing global health challenge receives so little public attention, while the mythology of Rh negative blood as evidence of alien ancestry attracts millions of engaged followers, is itself a revealing data point about how human attention and concern are distributed. The interesting and dramatic and personally meaningful story gets the attention. The quiet, persistent, entirely preventable tragedy gets ignored. This is not an accusation. It is a reflection of how human psychology works and of the specific ways that information environments shape what we notice and what we care about. But it is worth naming clearly. The most important thing about Rh negative blood, in terms of immediate human welfare, is not its possible ancient alien origins. It is the ongoing preventable mortality of infants whose mothers did not receive a simple preventive treatment during pregnancy. Chapter 11. The Identity Question. What blood type tells us about ourselves. Let's step back from both the science and the mythology for a moment and ask a more philosophical question. What does it mean to find your identity in your blood type? This is not a trivial question. The human need for identity, for a more coherent sense of who you are, where you came from, and what connects you to others, is one of the most powerful motivating forces in psychology. How we construct that identity, what materials we use to build it, and what consequences follow from different identity constructions are questions that matter deeply. Blood type identity is not a new phenomenon, and it is not limited to the Western Rh negative mythology. In Japan, the blood type personality theory, the belief that ABO blood type predicts personality characteristics in ways roughly analogous to astrology, is a mainstream cultural phenomenon, not a fringe belief. Surveys consistently show that a majority of Japanese people know their blood type and believe it influences personality. That blood type is a common topic of conversation in social and romantic contexts, and that hiring discrimination based on blood type, called burahara, blood type harassment, has been documented and is recognized as a social problem. The Japanese blood type personality theory has been subjected to extensive scientific scrutiny and has not held up. Large studies of personality and blood type have consistently found no statistically significant correlations between ABO blood type and any measured personality trait. The belief persists despite this lack of evidence, for reasons that are instructively similar to the reasons the Rh negative mythology persists. It provides a simple, memorable, socially shared framework for understanding human variation. It gives people a basis for forming social connections and expectations. And it is resistant to falsification because individual experience always seems to provide confirming examples, while disconfirming examples are easily dismissed. The broader phenomenon of blood type identity, the construction of personal and group identity around blood type, reveals something important about the relationship between biology and identity. We live in an era of increasing genetic awareness, in which direct-to-consumer genetic testing services have made it possible for millions of people to learn, in considerable detail, about their genetic ancestry, their carrier status for various genetic conditions, and their genetic relatives. This unprecedented access to biological information about oneself is changing the landscape of identity construction in ways we are only beginning to understand. The appeal of blood type identity is, in this context, understandable as a simplified version of a more general interest in biological self-knowledge. Blood type is simple, a clear category, a definitive result, something that does not change and does not require interpretation. Genetic ancestry is complex, probabilities and percentages and population categories that require considerable context to interpret, and that shift as reference databases are updated. Blood type is clean. The category is clear. You are either Rh negative or you are not. But this simplicity is, scientifically, a limitation rather than an advantage. Blood type is one locus, one small part of the genome, that tells you almost nothing about your overall genetic ancestry, your health predispositions, your personality, your intellectual capacities, or your connection to any particular historical lineage. The genome contains approximately 3 billion base pairs of information, encoding approximately 20,000 genes, and the 2 genes of the Rh blood group system represent a vanishingly small fraction of that total information. Inferring anything significant about identity from blood type alone is like inferring a novel's plot from a single letter. The Rh negative mythology by investing this single locus with enormous biographical and historical significance, is committing a form of genetic reductionism that the science of genetics explicitly warns against. Single genetic variants do not make you who you are. They are one tiny piece of a vast and complex information system, the overall pattern of which shapes biology in ways that cannot be inferred from any individual component. This is not to say that genetics is irrelevant to identity. It clearly is not, given how much of human variation in appearance, health, and even behavior has genetic components. But the relationship between genetics and identity is probabilistic, multifactorial, and deeply modulated by environment and experience. It is not the simple, determinate relationship that blood type mythology assumes. The healthiest relationship with biological self-knowledge is probably one that holds the information lightly, that finds it interesting without finding it determinative, that uses it as one input among many rather than as a master key to identity. Your Rh negative blood tells you something real and interesting about your red blood cells, about your medical profile, and very approximately about the ancient migrations of some of your distant ancestors. It does not tell you that you are special in a way that others are not. It does not tell you that you are descended from aliens or angels. It does not explain your sense of difference, or your difficulty fitting in, or your unusual perceptions. What it does tell you, and this is, in its own quiet way, remarkable enough, is that you are carrying a genetic variant that has been in the human lineage for hundreds of thousands of years, that was present in the people who painted the caves of Europe in the Ice Age, that persisted through the transitions from hunting and gathering to farming to the modern world, and that is expressed in your red blood cells at this particular moment in the ongoing story of the human species. That is not nothing. It is in fact quite something. It is just not aliens. Chapter 12, The Medical Frontier, Rh Blood, and the Future of Medicine. We are at an interesting moment in the history of blood-type medicine. The discovery of the ABO and Rh blood group systems in the 20th century made safe blood transfusion possible and prevented millions of deaths from Rh incompatibility. The development of Rh immunoglobulin was one of the great preventive medicine success stories of the century. But the scientific understanding of the Rh blood group system and its clinical applications is still developing, and some of the most interesting developments are quite recent. The immunology of Rh incompatibility is more complex than the simple sensitization and attack model that the clinical guidelines are based on. Research over the past decade has revealed that the immune response to Rh antigens involves multiple arms of the immune system, not just the antibody-producing B cells that are the target of Rh immunoglobulin, but T cells, regulatory immune cells, and innate immune mechanisms. Understanding this complexity has implications for improving both prevention and treatment of hemolytic disease. One of the most promising areas of current research involves the development of more targeted and more durable forms of Rh immunoglobulin. The current preparations are derived from human plasma donors who have been sensitized to the Rhd antigen, a process that requires finding and maintaining a pool of sensitized donors, which is becoming increasingly difficult as Rh immunoglobulin administration has become more widespread, and fewer Rh negative individuals are naturally sensitized. Recombinant anti-R, HD monoclonal antibodies, engineered proteins that mimic the activity of the natural antibodies in Rh immunoglobulin, have been in development for years and are showing promise in clinical trials. A successful recombinant product would eliminate the dependence on human plasma donors, improve consistency and purity, and potentially reduce costs, making it more accessible in low-resource settings. The genetic prediction of Rh blood type, determining blood type from DNA sequence rather than from traditional. Serological testing is another frontier with significant clinical implications. Traditional blood typing uses serological methods, mixing blood with antibodies and observing whether agglutination occurs. These methods are reliable but require specific reagents, trained technicians, and are subject to certain technical limitations. Molecular blood typing, determining the underlying genetic basis of blood type from a DNA sample, is increasingly accurate, cost-effective, and informative, particularly for patients who have received recent transfusions, which can confound serological testing, and for fetuses whose blood type needs to be determined before birth. Non-invasive fetal Rh blood typing, the determination of whether a fetus is Rh positive or Rh negative from a maternal blood sample using cell-free fetal DNA circulating in the mother's blood, is now clinically available in many countries and is transforming prenatal management of Rh incompatibility. In the DARF traditional approach, all Rh negative pregnant women receive Rh immunoglobulin prophylactically, even those carrying Rh negative fetuses who are not at risk. Non-invasive fetal typing allows the identification of the approximately 40% of Rh negative women who are carrying Rh negative fetuses and who do not need Rh immunoglobulin, reducing unnecessary treatment and the associated use of a scarce blood product. The broader field of blood group genomics, the systematic molecular characterization of all blood group antigens and their genetic basis, has advanced dramatically over the past decade. The complete Rh blood group system, with its dozens of antigens and complex genetic variants, has been molecularly characterized in detail that was impossible even 20 years ago. This detailed molecular knowledge is enabling more precise matching of blood donors and recipients for transfusion, reducing alloimmunization and improving outcomes for patients who require repeated transfusions, including patients with sickle cell disease, thalassemia, and other conditions requiring chronic transfusion support. Extended blood group matching, matching donors and recipients not just for ABO and RHD, but for a broader panel of blood group antigens, is standard of care for some patient populations and is becoming more widely practiced. The molecular approach to extended matching is more accurate and more scalable than traditional serological typing, and ongoing work is making it more affordable and more accessible for routine clinical use. The potential connection between Rh blood type and disease susceptibility, the research into toxoplasma, and other potential associations that we mentioned in the previous section, remains an active area of inquiry. If genuine associations are established between Rh blood type and susceptibility to specific infectious diseases, this could have implications for understanding why different Rh blood type frequencies evolved in different populations, for identifying individuals who might benefit from particular preventive strategies, and for understanding disease mechanisms. The research into the functional role of Rh proteins in red cell physiology, their potential role in gas transport, ion channel function, and other aspects of red cell biology continues in parallel with the clinical and population genetic research. Understanding what the Rh proteins actually do in the red blood cell membrane might reveal why the D antigen specifically is so immunogenic, why the immune system of an Rh negative individual responds so strongly to Rhd antigen, more strongly than to most other blood group antigens. This immunogenicity is clinically important but mechanistically not fully explained, and understanding it better could lead to new approaches to preventing Rh sensitization. The future of blood type medicine is one of increasing precision, more detailed molecular characterization, more targeted clinical interventions, more sophisticated understanding of the immunological mechanisms involved, and better global access to the preventive treatments that already exist. The fundamental discovery made by Landsteiner and Levine and Wiener in the 1930s and 1940s that Rh blood type matters clinically and matters in ways that can be addressed medically remains the foundation of everything that follows. What is being built on that foundation is an increasingly detailed and increasingly powerful structure of knowledge and clinical capacity. None of this requires aliens, but all of it is genuinely extraordinary. Chapter 13. The Ancient Civilizations. Egypt, Sumer, and the Blood of Kings. You are standing in the Valley of the Kings. The heat is extraordinary, the kind that makes the air itself seem to vibrate, that turns the limestone cliffs into something almost hallucinatory in the afternoon light. Around you, cut into the rock faces of this narrow canyon on the west bank of the Nile, are the entrances to tombs that were already ancient when Julius Caesar visited Egypt. Tombs of pharaohs, tombs of the divine kings who ruled the most powerful civilization of the ancient world for 3,000 continuous years. And somewhere in the rock beneath your feet, in the preserved, carefully mummified remains of those divine kings, is biological information that scientists have only recently begun to extract and analyze, including, among many other things, blood type data. The mummification practices of ancient Egypt preserved biological material with a thoroughness that continues to astound modern researchers. Not just the gross anatomy, the bones, the organs, the skin, but the microscopic material. Cells, proteins, DNA fragments, and in some well-preserved specimens, the antigens on red blood cell membranes that determine blood type. The analysis of blood type antigens in ancient Egyptian mummies has been attempted since the mid-20th century, with variable success depending on the preservation state of the specimen and the sophistication of the analytical methods available. The results of this research are genuinely interesting, and they have been enthusiastically and often inaccurately incorporated into the Rh negative mythology. Let's start with what the science actually found. Studies of ABO blood type in ancient Egyptian mummies, conducted by researchers at various institutions from the 1970s onward, found ABO blood type distributions in the ancient Egyptian population that were broadly similar to those found in modern Egyptian and North African populations. Type A and Type O were both common. Type B was present. The distribution was consistent with what population genetics would predict for a North African population, with connections to both sub-Saharan Africa and the Middle East, which is, of course, exactly what the ancient Egyptians were. The Rh blood group is harder to analyze in ancient material than ABO, because the Rh antigens are more fragile and more susceptible to degradation over time. The Rhd antigen in particular, the antigen whose presence or absence defines Rh positive or Rh negative status, has been analyzed in some well-preserved mummy specimens with mixed results. Some studies have reported finding Rhd antigen in some ancient Egyptian samples, consistent with Rh positive blood. Others have found specimens that appear to be Rh negative. The overall picture, such as it is, suggests that ancient Egyptians had a distribution of Rh blood types roughly consistent with modern North African populations. That is, predominantly R positive with minority of Triar, Rh-negative individuals at frequencies similar to modern North African populations. This is not, it should be noted, a high Rh-negative frequency. North African populations today have Rh-negative frequencies, somewhat higher than sub-Saharan African populations, but substantially lower than European populations, typically in the range of 5 to 10%. The ancient Egyptian data, fragmentary as it is consistent with this range. The mythology, predictably, has interpreted these findings differently. The claim that ancient Egyptian royalty had high rates of Rh-negative blood, specifically that the pharaohs were Rh-negative, that this is why they insisted on marrying within their own family, appears repeatedly in Rh-negative mythology, literature, and online communities. Let's examine this claim carefully. First, the genetic analysis that has been performed on ancient Egyptian royal remains has focused primarily on autosomal DNA, the genetic material that determines ancestry, rather than specifically on blood group loci. The most famous ancient DNA study of Egyptian royal remains was the 2010 analysis of Tutankhamun's family, which revealed a pattern of close inbreeding, consistent with the known practice of brother-sister and other close kin marriages in the royal family, and identified certain hereditary conditions in Tutankhamun's mummy. This study did not specifically characterize Rh blood type. Second, the Egyptian royal practice of close-kin marriage, while real and documented, was motivated by theological rather than genetic reasons, as we discussed in the previous Egyptian pharaoh's script. The pharaoh married his sister or close relative because the theology of divine kingship required the concentration of divine blood, not because of any awareness of blood type compatibility. The connection between Egyptian royal endogamy and Rh blood type is an invention of the mythology, not a finding of the archaeology. Third, whatever the Rh blood type frequency in ancient Egyptian royalty was, we currently lack the data to characterize it with confidence. The ancient DNA and blood type analysis of Egyptian royal mummies is ongoing, and new studies using improved molecular techniques are periodically published, but the existing data does not support the specific claim that Egyptian pharaohs were predominantly or exclusively Rh negative. The mythology's interest in ancient Egypt and Rh blood type is, however, connected to a genuine and interesting observation about ancient Egyptian civilization, that it produced, over its 3,000-year span, a remarkable concentration of intellectual, artistic, and administrative achievement that has prompted questions about what made it possible. The pyramids, the temples, the administrative systems, the medical knowledge, the astronomical observations, the achievements of ancient Egypt are genuinely extraordinary, and it is understandable that people look for explanations. The Rh negative mythology offers the explanation that the Egyptian elite, or at least the royal family, carried special blood that enabled these achievements. The scientific explanation is considerably more mundane, but no less interesting. Ancient Egyptian civilization's achievements reflect the accumulated knowledge of many generations of skilled, intelligent human beings working within a stable and well-resourced institutional framework. The pyramids were not built by special blood. They were built by organizational genius, mathematical knowledge, and tens of thousands of well-fed and reasonably well-treated workers, as the archaeological evidence of the workers' village at Giza makes abundantly clear. Now let's travel further east and further back in time to the other ancient civilization that appears most frequently in Rh negative mythology. Sumer, the Sumerian civilization of ancient Mesopotamia, the region between the Tigris and Euphrates rivers in what is now Iraq. is among the earliest complex civilizations in the archaeological record. The Sumerians developed one of the first writing systems, built the first cities, established the first formal legal codes, and created a mythology that has influenced Western religious tradition to the present day. The Sumerian texts, tens of thousands of clay tablets inscribed with cuneiform writing, preserve an extraordinary body of literature, administrative records, astronomical observations, and mythological narratives. The Sumerian texts that are most relevant to Rh- mythology are the ones describing the Anunnaki, the great gods of the Sumerian pantheon. In the Rh- mythology, as filtered through the work of Zecharia Sitchin and his successors, the Anunnaki are not mythological beings, but actual extraterrestrial visitors who came to Earth from a planet called Nibiru, genetically modified existing hominids to create modern humans, and then interacted with their human creations in ways recorded in the Sumerian mythological texts. The Rh- blood connection enters here in the form of the claim that the Anunnaki, as extraterrestrials, would have had blood that was incompatible with ordinary human blood, and that their hybrid offspring, the beings who became the royal and priestly elites of early Sumerian civilization, carried this incompatible blood as a genetic legacy. The Rh- condition, specifically the immune incompatibility between Rh- mothers and Rh-positive fetuses, is interpreted as the biological trace of this hybrid ancestry, the Rh- mother's body, recognizing that her Rh-positive fetus carries the contaminated human blood rather than the pure, ancestral, extraterrestrial blood. The Sitchin hypothesis, the interpretive framework from which this mythology derives, has been extensively critiqued by actual scholars of ancient Mesopotamian languages and culture. Sitchin's translations of Sumerian texts are, according to professional Assyriologists, not translations at all. They are creative reinterpretations that bear little relationship to what the texts actually say when read by scholars with genuine expertise in cuneiform, Sumerian, and Akkadian. The specific claims Sitchin makes about Nibiru, about genetic modification of hominids, about the Anunnaki as extraterrestrials. None of these are supported by the actual content of the ancient texts, as understood by scholars who read them in the original. This is not a matter of scholarly interpretation, of different experts reading the same evidence differently. It is a matter of linguistic competence. People who can actually read Sumerian cuneiform say, consistently, that the texts do not say what Sitchin claims they say. This is a fairly definitive refutation of an argument that depends entirely on what the texts say. The genuine archaeology of Sumerian civilization is, once again, remarkable enough on its own terms. The development of writing, the cuneiform script that allowed Sumerian scribes to record not just inventories and administrative records, but literature, mythology, law, and scientific observation, is one of the most consequential developments in human history. The Epic of Gilgamesh, preserved in cuneiform tablets, and representing one of the earliest works of literary narrative we have, explores themes of mortality, friendship, and the limits of human ambition with a sophistication that continues to resonate 4,000 years after it was written. The Sumerians did not need extraterrestrial assistance to achieve what they achieved. They needed the specific combination of geographical circumstances, the fertile floodplain of Mesopotamia, the rivers that allowed trade and communication, the climate that supported agriculture, and human intelligence applied systematically over many generations. The result was a civilization of extraordinary achievement. That achievement belongs to them and to their humanity, not to alien genetic engineers. The blood type data from ancient Mesopotamian populations is even more fragmentary than the Egyptian data. The preservation conditions in Mesopotamian archaeological sites are generally less favorable for biological material than in Egypt, and what exists does not support the specific claims made in the mythology. The ancient populations of Mesopotamia appear to have had blood type distributions broadly consistent with modern Middle Eastern populations, with no particular evidence of extraordinary Rh negative frequencies. The pattern across both Egypt and Sumer is consistent. The mythology selects ancient civilizations of demonstrated achievement, proposes that their achievements required blood that was different from ordinary human blood, and then reads that blood difference backward onto whatever biological data can be made to fit. The actual biological data, when it exists, does not support the specific claims. The actual achievements of these civilizations, when examined historically, are entirely explicable in human terms. But the mythological framework persists because it is meeting needs that the historical explanation does not meet. The need for a sense of ancient connection to greatness, for a lineage that links the individual directly to the pyramids and the ziggurats and the written word, is a powerful need. And the blood type provides the biological hook on which that connection can be hung. The reality is that every human being alive today is descended from the people who built the pyramids, not specifically the pharaohs, but the population of ancient Egypt, in the same sense that every human being is descended from all surviving human lineages of that period. The connection is real. It is just not mediated specifically by Rh negative blood. It is mediated by the simple fact of human continuity across time, the unbroken chain of reproduction that links every living person to every human who lived 10,000 years ago. That is not a special bloodline. It is the human lineage. All of it shared by everyone. Chapter 14, The Immune System Advantage. Disease, resistance, and the hidden biology of Rh negative blood. Here is something that the mythology gets approximately right for entirely the wrong reasons. Rh negative blood may actually confer some biological advantages in specific contexts. Not the advantages the mythology claims, not psychic powers, not alien resilience, not immunity to the control mechanisms of the reptilian elite, but real, measurable, biologically plausible advantages in specific disease contexts that researchers have been investigating with increasing sophistication. The investigation of blood type and disease susceptibility is a legitimate and active area of biomedical research. The ABO blood group system's connections to disease susceptibility are the best established. Type O individuals have well-documented partial resistance to severe malaria caused by Plasmodium falciparum, because the malaria parasite uses the A and B antigens on red blood cells as attachment points, and cells lacking. These antigens are harder for the parasite to invade and use as a vehicle for rosetting. The clumping of infected and uninfected red blood cells that contributes to the most severe manifestations of malaria. Type A individuals have elevated susceptibility to certain bacterial infections. Type AB individuals have elevated risk of certain cardiovascular conditions. These are statistically real effects, mechanistically understood at least in broad outline, and clinically relevant in populations where the relevant diseases are endemic. They represent genuine natural selection operating on blood type variants, genuine evolutionary pressure that has shaped the global distribution of ABO blood type frequencies in ways that correlate with the geographic distribution of specific pathogens. The question of whether analogous effects exist for the Rh blood group system, whether Rh positive or Rh negative status confers any advantage or disadvantage in specific disease contexts, is less well studied but has attracted increasing research attention, and the results, while preliminary and sometimes contested, are genuinely interesting. The most extensively studied potential association between Rh blood type and disease involves the parasite Toxoplasma gondii. Toxoplasma is a protozoan parasite that infects a very large fraction of the global human population. Estimates suggest that somewhere between 30 and 50% of people worldwide carry a latent Toxoplasma infection. The parasite is transmitted primarily through contact with cat feces and through consumption of undercooked meat from infected animals. In healthy adults with intact immune systems, the infection is typically asymptomatic or causes only mild flu-like symptoms. The immune system contains the parasite, which then enters a latent phase in muscle and brain tissue. However, latent Toxoplasma infection has been associated in some research with subtle effects on behavior and psychology. Studies by Czech researcher Jaroslav Fleger and others have found associations between latent Toxoplasma infection and differences in reaction time, risk-taking behavior, and certain personality traits. The mechanism proposed involves the parasite's manipulation of dopamine pathways in the brain. Toxoplasma, like many parasites, appears to be able to influence host behavior in ways that might favor its own transmission. The connection to Rh blood type enters through research suggesting that the behavioral and psychological effects of Toxoplasma infection differ between Rh positive and Rh negative individuals. Studies have found that some of the behavioral associations with Toxoplasma infection, particularly effects on reaction time and certain personality measures, are statistically significant in Rh positive individuals, but attenuated or absent in Rh negative individuals. This has been interpreted as suggesting that Rh negative individuals may be partially resistant to Toxoplasma's neurological effects, possibly because the Rhd protein plays some role in the parasite's interaction with host cells. This research is preliminary, has been conducted primarily by a small number of research groups, and has not been consistently replicated. The effect sizes reported are small. The mechanistic explanation is speculative. It is not known whether or how the Rhd protein would be involved in Toxoplasma's neurological effects, and the overall interpretation is complicated by the many confounding factors that complicate research on behavioral associations with parasitic infection. But the research is not pseudoscience. It is published in peer-reviewed journals, conducted with appropriate statistical methods, and generating ongoing scientific discussion. Whether the Rh blood type genuinely modifies susceptibility to Toxoplasma's effects is an open scientific question, and open scientific questions are the most interesting kind. Beyond Toxoplasma, researchers have investigated potential associations between Rh blood type and a range of other conditions. Some studies have found associations between Rh negative status and differences in immune function, specifically in the balance between different types of immune responses. The immune system has two broad modes of operation: the Th1 response, oriented toward fighting intracellular pathogens like viruses and certain bacteria, and the Th2 response, oriented toward fighting parasites and also implicated in allergic responses. Some research has suggested that Rh-negative individuals may have a tendency toward a slightly different balance between these modes, though the data is inconsistent and the effect sizes are small. Associations between Rh blood type and autoimmune conditions have also been investigated. Autoimmune diseases, conditions in which the immune system attacks the body's own tissues, have complex genetic underpinnings, and blood type variants are among the many genetic factors that have been examined as potential risk modifiers. Some studies have found weak associations between Rh blood type and certain autoimmune conditions, but these associations are not consistently replicated and are not large enough to be clinically useful for predicting individual risk. The connection between Rh blood type and susceptibility to specific viral infections has attracted research attention, particularly since the COVID-19 pandemic, when the association between ABO blood type and COVID-19 susceptibility and severity was reported in several large studies. The finding that type O individuals had somewhat lower susceptibility to SARS-CoV-2 infection, and that type A individuals had somewhat higher severity of disease, prompted investigators to look at whether Rh status also influenced COVID-19 outcomes. Several studies examined the association between Rh blood type and COVID-19 with mixed results. Some found that Rh-negative individuals had modestly lower rates of COVID-19 infection or slightly less severe outcomes, while others found no significant association after controlling for confounding factors. The overall picture is inconclusive. The effect of Rh blood type on COVID-19, if any, appears to be small and inconsistently demonstrable across different study populations and methodologies. The honest assessment of the current research on Rh blood type and disease is there may be real biological effects of Rh-negative status on disease susceptibility and immune function, but they are small, inconsistently demonstrated, and not yet mechanistically explained. The research is active and ongoing. New findings are being generated. Some of those findings will likely replicate and eventually be established as genuine biological effects. Others will turn out to be statistical noise or artifacts of confounding. This is how science works. It is slow, cumulative, self-correcting, and ultimately more reliable than any alternative for getting at biological truth. The fact that the science is uncertain and ongoing is not a weakness of the scientific approach. It is an honest reflection of the genuine complexity of biology. What the research does not support is the mythology's strong version of Rh-negative advantage, that Rh-negative individuals are substantially more resistant to disease in general, that they have superior immune systems, that they are physically hardier or more resilient than Rh-positive individuals. The effect sizes in the research literature are modest. They might, on a population level, influence the frequency of certain diseases in groups with different Rh-type distributions, but they would not be noticeable at the level of individual experience. The Rh-negative person is not, by virtue of their blood type, meaningfully more disease-resistant than their Rh-positive neighbor. The one clear biological difference between Rh-negative and Rh-positive individuals, the Rh incompatibility issue in pregnancy, is a disadvantage for Rh-negative individuals in the specific context of carrying Rh-positive fetuses. This disadvantage is real, historically significant in terms of infant mortality, and only fully addressed by modern medicine. It is, as we have emphasized throughout this video, entirely the opposite of what the mythology claims. Not a sign of superior ancient lineage, but a medical complication requiring active management. The immune system of Rh-negative individuals is not, in any documented sense, generally superior to that of Rh-positive individuals. It is differently configured in specific and subtle ways that may have specific and subtle consequences in specific disease contexts. This is interesting biologically. It is not the extraordinary advantage that the mythology claims, but here is what is worth taking seriously from the research. The Rh blood group system is not immunologically inert. The Rhd protein is a real molecule with real biological functions beyond its role, and as a blood group antigen, the immune system's response to Rh antigens is real and powerful enough that it can destroy fetal red blood cells across the placental barrier. And the interactions between Rh blood type and the broader immune system, while incompletely characterized, are a legitimate area of active research. The full biology of the Rh blood group system, its evolutionary history, its physiological functions, its interactions with pathogens, and with the immune system, is not yet completely understood. The story is genuinely not finished. New research is adding new chapters all the time, and some of those chapters may reveal biological significance that current understanding does not anticipate. This genuine openness, the honest acknowledgement that there is more to learn about Rh blood type biology than is currently known, is sometimes seized on by the mythology as license for any claim whatsoever. If science doesn't fully understand it, then maybe the aliens are right, but this is not how epistemic humility works. The fact that we don't know everything about a subject does not mean that all claims about it are equally valid. The fact that Rh-negative biology is incompletely understood does not make extraterrestrial ancestry a reasonable hypothesis. It makes continued rigorous scientific research the appropriate response. The biology of your blood type is genuinely not fully understood. The science is actively working on it. The answers it finds will be biological answers about proteins and immune cells and evolutionary pressures and disease mechanisms, grounded in evidence and testable by experiment. Those answers will be interesting. They will not be aliens. They will be something better, the actual complex, still being discovered truth about what your blood is doing in your body and why. Conclusion: the blood and the story. Let's come back to where we started, the doctor's office, the blood test, the 2 letters and the symbol, Rh negative. You know much more now than you did at the beginning of this video. You know what those letters mean in molecular terms. The absence of a specific protein on the surface of your red blood cells, encoded by the Rhd gene, which is either deleted or non-functional in people who are Rh negative. You know what they mean in evolutionary terms, a genetic variant that is hundreds of thousands of years old, that was present in the early European hunter-gatherers who painted the caves of Altamira, that persisted through the population turnovers and migrations of the Neolithic and Bronze Age, and that survives today in its highest frequency in the Basque people. The living inheritors of Europe's oldest genetic legacy. You know what they mean in medical terms. A blood type characteristic that requires specific consideration in transfusion and in pregnancy, that has been the source of enormous infant mortality in the past, and continues to cause preventable deaths in parts of the world that lack access to Rh immunoglobulin. And that was the subject of one of the 20th century's most consequential medical discoveries. You know what they mean in the mythological ecosystem of the internet. a mark of special ancestry, of ancient alien lineage, of the Nephilim or the Atlanteans, or some other non-ordinary human origin, a biological badge of difference that explains why you have always felt somehow apart from the ordinary world, and now you have to decide what the two letters mean to you. Here is what I would suggest for whatever it is worth. The genuine story, the real, scientifically grounded, evidence-based story of Rh-negative blood is remarkable enough to not need embellishment. The idea that you are carrying in your red blood cells a genetic variant that was present in the people who survived the last ice age in the mountains of Western Europe, who watched the glaciers retreat and the forests spread, who hunted the animals and gathered the plants of a world that no longer exists. This is extraordinary. These were fully modern human beings with minds and emotions and social lives as complex as ours, living in landscapes we can barely imagine, and their genetic legacy is in your blood. The Basque people, who carry this legacy most strongly, have maintained their linguistic and genetic distinctiveness across the entire span of recorded European history, through the Roman conquest, through the Visigothic kingdoms, through the Frankish Empire, through the development of the Spanish and French nation-states, through every political and demographic upheaval of the past 2,000 years. They are still there. They still speak their ancient language, and in their blood, the highest surviving frequency of Rh negative in the world marks them as the most direct living link to the pre-agricultural Europeans who were the first people we might call, in any meaningful sense, Europeans. That is not a story about aliens. It is a story about human persistence, about the continuity of human communities across enormous spans of time, about the way that genetic information carries the history of our species in every cell of our bodies. It is a story that the actual science is making clearer with every new ancient genome analyzed, every new population study published, every new piece of the prehistoric human puzzle put into place. The mythology, the alien bloodlines and the Nephilim, and the suppressed secrets offers something different. It offers the feeling of special significance, of belonging to a lineage that matters cosmically rather than merely historically. It offers a community of people who share your blood type, and through that sharing, a sense of connection and identity. It offers an explanation for the feeling of being different that is flattering rather than merely descriptive. These are real human needs, and the mythology meets them effectively. But it meets them with a story that is false, and the falseness matters. Not because being wrong is a moral failure, but because the false story substitutes an invented drama for a real one, and the real one is better. The real story is that you are a human being carrying biological information that connects you to every other human who has ever lived, to the long chain of ancestors who survived long enough to reproduce, going back through all of recorded history and beyond, through the Ice Age and the first emergence of anatomically modern humans in Africa, and the evolutionary lineages that preceded them. Your blood type is one tiny part of that biological information, reflecting one small corner of the deep history of our species. It is not proof of alien ancestry. It is not the mark of the Nephilim. It is not a secret that the powers of the world are suppressing. It is a piece of your inheritance from the full, extraordinary, entirely human story of how we got here. And that story, the real one, is more than enough. You close the browser. You put away the 17 tabs. You hold the blood test result in your hands for a moment, with all of its genuine meaning and none of its invented mythology, and you recognize it for what it is, a small window into a very long story. Your story, the human story, the only one we have, and the only one we need